Thank you very much for Professor Dux to introduce me. I have joined the program as one of the first mentors in 2013. At that time, it was called Szeged Scientists Academy, and now from this year, it's a National Scientists Academy. I am heading the Biological Barriers Group, and I would like to give an overview about what are the barrier systems and what are they good for. So our body is protected on many levels. So we have outer biological barriers. These are the epithelial barriers, like the skin, like the very outer layer of the eye, which we call cornea epithelial layer. We also have barrier system in our airways, starting from the nasal system down to the lungs. And also we have the gastrointestinal epithelium, and all these layers are separating our body from the outside environment. We also have some inner barriers, like the blood-brain barrier in our head. Also, we have some specialized barrier systems like the placenta between fetuses and the mothers and others. Now, the barrier systems that are indicated in red color are the ones that my team is interested in and have been working with models. The major role of this barrier is having a protection against possible harmful effects. This is done through physical barriers, and also we have chemoprotection. Physical barriers is basically made by junctions between cells. This is what we call intercellular junctions, and you can see that the two cell leaflets are joined by these yellow representation of actual protein molecules. And they are stopping, for example, bacteria or viruses to getting into the space between the cells. We also have some protein molecules that act as pumps, which are catching harmful molecules, and then they are effluxing them, pushing back, and don't let them to enter the body. These protection systems, however, are limiting drug entry to the body as a whole and to different organs. And this limited entry is especially true for water-soluble molecules like nutrients and a lot of biotherapeutics that are increasingly used in the therapy of diseases. And also we have many, many drugs like cancer drugs and antibiotics, so antibacterial, antiviral drugs that are not let to enter the body by these efflux pumps. So this is the reason why we have difficulty to treat some neurological diseases, or we cannot get every kind of drug through oral pills, but only for injections. And also barriers are important because we know now that many diseases are damaging this kind of protective barrier. And of course, if these are diseases of the brain, it seems logical, like Alzheimer's, Parkinson's, epilepsy, brain tumors, or stroke. However, it's not well known that systemic diseases can also have a bad effect on the barriers, and that includes like alcoholism or diabetes or many infectious diseases like the neuro-AIDS, which is caused by the HIV virus. So how to study these barriers? We are lucky because we are using some very ingenious little tricks. For example, culture inserts, which are made from sterile plastic cylinders with a porous membrane, are perfect for cell culturing. And this way we have a confluent monolayer of cells separating two fluid compartments. And we can measure the integrity of the barrier by measuring electrical resistance, and we can also study the permeability of molecule through these systems. One of the models that was developed by our team, together with Japanese partners, is a triple co-culture model that is mimicking the anatomical structure of brain capillaries forming the blood-brain barrier. So we are having endothelial cells on the upper side of these porous membranes. We have pericytes on the bottom side of the filter membrane, and finally, the third cell type, glial cells, is cultured at the bottom of the culture plates. And this system is basically a very useful model system to study drug transport and diseases. We further developed these kind of culture insists, and we were introducing with our biophysicist colleagues a microfluidic and electronic chip device, which is two microchannels with a porous membrane in the middle and sandwiched between two slides, having on the inner side a very thin layer of gold electrodes. And this is the model we used for mimicking the blood flow in the system. So in contrast to the static system, now we have a system that's dynamic, and we also have flow as an additional physiological factor. And we basically introduced a human blood-brain barrier model using endothelial cells and brain pericytes, and we could show that such a model has got better resistance and lower permeability, meaning tighter barrier, making it more close to what we have in our brains. So the two major research areas my team is, one is like drug delivery across barriers, and for this we use targeted drug delivery with nanoparticles, and the second, we are studying how the blood-brain barrier is damaged in diseases and how we can protect this barrier in these disease types. Currently, we work on Parkinson's disease, but we previously worked on epilepsy, and we also worked on pancreatitis. Just one slide about our nanoparticle research, which is spanning now about almost 10 years. And one of my students, Gergő Porkola, participated in this nanoparticle study. So we decorated the surface of nanoparticle with different nutrient ligands. And in addition, because we know that the barriers express many types of nutrient transporters, we did combination of ligands, combination of molecule on the surface. And what we could show using the previously introduced co-culture model of the blood-brain barrier, we could demonstrate that nanoparticles that are having combination of ligands on their surfaces, namely alanine, the amino acid, and glutathione, the tripeptide, is crossing much better this model than the cargo of the nanoparticle or the undecorated nanoparticles, or even those nanoparticles that has just one ligand on their surface. We are now working on a project when we are not only combine different nanoparticulate drug delivery systems and cheap devices with barriers, but we are having also midbrain organoids, which are, I would say, mini brains. These are about 1 millimeter size, but they are spheroids and having neurons and glial cells. And we are now having a complex system in which we not only study that how the nanoparticles can cross the barriers, but do they reach the mini brains and are able to enter them and possibly correct the disturbances which is found in diseases. most importantly, this is an international cooperation between teams in Austria, Taiwan, Luxembourg, Israel, and Switzerland. And that's my biological barriers research group, so if you are interested, you can see and check our page on Facebook. And my last slide, I would like to present you my students. So I am very proud to be able to work with Gergő Porkoláb, who finished his grammar school here in Szeged. He went to the István Tömörkény Grammar School, and his teacher was István Csigér, who is also, we are very proud in this network of biology teachers who is helping the foundation. And after Gergő spent a summer student practice with my team, he decided to join it, and he became a Szent-Györgyi student, and during his years, he really progressed very well. So he got a lot of prizes, and we are very proud because he's the first PhD student in the National Scientist Academy. And this year, 2 more students joined my team. Anna Hegyi finished her secondary school studies in Miklós Radnóti Grammar School, and Evese Pál, she finished in Kálmán Könyves Grammar School in Újpest. And both of them are studying medicine at the University of Szeged, and both of them started in 21 January their long, hopefully long trip in the field of research, biomedical research. With that, thank you very much for your attention.
Thank you very much for this very stimulating presentation. And now we have a couple of minutes for questions. So I encourage you to come forward, raise your hand, and if you want to ask something from Professor Deli, just don't be shy. This is one of the most important and most valuable part of these conferences, that the students can ask questions or make comments. Is there anybody ready now? Yes, one of our students.
Thank you. I have to ask, because I think it is a very exciting topic, and probably you know very well the recent paper by the group of Tolibis-Corey about the increased penetration of different plasma proteins into the brain across the BBB as opposed to our belief. And certainly this transport changes a lot by aging. So how do you feel this changes the landscape, in a sense that we don't just have to think about the BBB as a barrier, but we have to also study how many things get across as default?
Yeah, I think we need to study both. So what is the most intriguing related to proteins and plasma proteins? So not all of them are able to penetrate, and they are penetrating in a different extent. So albumin, which makes most of the blood protein, it's dangerous for the brain because of 2 reasons. One, when albumin crosses, I would say, brain capillaries, let's name it, it almost always goes with water transport too. And that makes brain edema. This is one of the reasons why the albumin transport across brain capillary endothelial cells is very low. And second, once albumin gets across, it is becoming in contact with astrocytic end feet. And actually, albumin is triggering astrocytes to become neuroinflammatory. And that's very well described in the work of Professor Alan Friedman. And this is the reason, for example, why we can observe somebody, I would say, we have a secondary school student who is rollerblading or doing skating or something, and is falling down and hitting the head. And this student is fine, but let's say 2-3 months after this head injury, he or she develops epilepsy. And the reason is that in the original trauma to the brain, there's a local, very mild blood-brain barrier opening, which is focally lets albumin to enter the brain. So a neuroinflammation starts that will change the glial cells, that will initiate this neuroinflammation, that will result in the seizure. And if it's not treated, this first seizure can go into a cycle, resulting in maybe really in epilepsy as a disease, which has like very serious consequences. Well, we know that the brain is not hermetically closed for proteins, and some proteins do cross. For example, especially in the neonates, we have the so-called FC receptor, which is able to bind immune globulins and transfer it across. We also know that, for example, in diseases and aging, these protective systems and mechanisms change too. So while in young people, we don't see, for example, fibrinogen leakage to the brain in young and healthy circumstances, in aging or disease, it may appear. And that has also, again, tremendous consequences, because this perivascular deposition of these molecules is really not making good things, unfortunately.
Thank you very much. Is there any question in the meantime you would like to ask Professor Deli? If not, then thank you very much again.
Thank you so much.